Good morning, everyone, and welcome to myDNAhealth's webinar on inflammation and the role of epigenetics and gene expression. It's 11 a.m. and we are ready to start, so thank you for joining us this morning. Those of you who have already attended our webinars, you know the drill. But for those of you who are joining us for the first time, just a little bit of housekeeping. We will send out the full webinar recording to all those who have registered, so don't worry if you miss anything or if you can't stay for the full session. This webinar is BANT and NNA accredited for CPD, so if you would like your certificate of attendance, just drop us an email and we will send it your way. We want this webinar to be as interactive as possible, so don't be shy and ask your questions all throughout the session using the chat box on your left-hand side. If you are feeling shy, you can always choose to send the question just to presenters and we will make sure to answer your question. Many of you may have already met our presenter, Dr. Shania Lee. But for those of you who haven't, Shania has been a doctor of complementary and alternative medicine since 2000 and has over a decade of experience of using genetic testing in her clinical practice alongside advanced functional medicine. So you are definitely in good and very capable hands this morning. The session is scheduled to run for 50 minutes, after which we will have 10 minutes of questions and answers. So to move on to our agenda this morning, we will learn what inflammation is and how it works, types of inflammation, genetics and epigenetics of inflammation, and finally, what you as a practitioner can do to help your clients that may be feeling inflamed. But before we start, a quick poll for you. So is all inflammation bad? Yes or no? Tell us what you think. Okay, I can see the results coming in, and most of you think no, inflammation is not all bad. Okay, thank you for that. So we can start our presentation now, and over to you, Shania.
Good morning, everyone. I'm really pleased to see the answers to that poll. Thank you all for taking the time out of your day to attend this webinar. I'm going to cover the mechanisms of inflammation and then go into the 3 genes that we test, as well as the epigenetic triggers and mediators of inflammation. I'm going to explain why we at myDNAhealth have included the specific SNPs we test, as well as including the questionnaire. And my hope is that by the end of this session, you will feel more confident when reading your clients' reports. So the answer to the poll is absolutely no. Inflammation is in fact a very necessary step in our innate healing response. It's through the inflammatory response that we can remove potential pathogens, as well as remove damaged or dying tissue so that we ourselves can regenerate and heal effectively. Essentially, it's a vital and necessary process for survival. It's a protective and natural immunological process that assists with repair and recovery. So if inflammation is doing its job properly, there are 5 R's, and we like our 5 R's in this profession, so I'm going to go through the 5 R's of inflammation. We have the recognition of the injurious agent, and that can also include not just a pathogen, but also, say, an old and dying tissue cell of our own. It includes the recruitment of the white blood cells, the removal of whatever needs to be removed, the regulation of the inflammatory response, and this is where things get a little bit tricky and where the problems with inflammation come in, because our ultimate goal is resolution. If we don't have resolution of the inflammatory response, healing has not taken place and the condition becomes chronic, and that's when the problems occur. So another way of looking at this is that inflammation serves to dilute the inflammatory agent, whatever it is. And there's a saying in functional medicine that the solution to pollution is dilution. So that's where that would come in. It looks to destroy or neutralize the infectious or inflammatory agent so that healing and repair can take place, with the ultimate goal being restoration of function. So it is very vital, and you can imagine what your life would be like without an active inflammatory response. The obvious signs of inflammation, the pillars that we get taught when we're learning about inflammation at college, we have the redness and heat, pain and swelling, and sometimes loss of function. All of these are part and parcel of the vascular and cellular response that I'm going to be talking about in more detail. The loss of function is generally, you don't want to be moving an inflamed area or banging an inflamed area repeatedly. So it's a protective response, that one there. But inflammation is a double-edged sword. Okay, that's where it gets its bad reputation from. We know the benefits of inflammation, but the good does sometimes go bad. So inflammation, when it gets out of hand, can lead to many of what we know as chronic conditions, such as autoimmunity, cancer, serious cardiovascular conditions. In fact, those cardiovascular conditions, that's the main reason that drug companies spend millions each year investigating how they can block the certain inflammatory cytokines in order to prevent cardiovascular disease. That's how major a role they play in it. The focus of the pharmaceutical companies is predominantly on TNF-alpha and interleukin-6, which I'm going to be discussing shortly as well. But there are links at the bottom of each of the slides which do cite the research that I refer to. So when you get the slides, please take the time to actually read through it and educate yourselves on what's going on in the world. So let's review. I think all of us in practice and those students who are attending, prepare yourself for this, because these conditions are probably the most prevalent chronic conditions that we have right now. 20, 30 years ago, they weren't as prevalent as they are now, but now we've got pulmonary diseases, cancer, cardiovascular disease, Alzheimer's. I'm going to add dementia in there as well. It is an inflammatory disease. Type 2 diabetes, arthritis, and a big one being autoimmune disease. I mean, now we can have not just one autoimmune disease in a body, but multiple autoimmune diseases in a body. And also neurological disorders. Nobody wants to lose their neurological faculties. So essentially, if an inflammatory response has become basically gotten out of hand, we can look at the fact that there are genetic predispositions. That is a component, definitely. But it's also mediated by ongoing epigenetic influences, those things that are outside of ourselves influencing us. Things like poor diet, stress, bad sleep, and I'm going to go into those in more detail. So let's look at the two types of inflammation. The acute inflammation is, I think, the more obvious one. You bang your knee, you have a stimulus, you get some bruising, healing happens. It's short and sweet, and it generally resolves itself with a healing. Unfortunately, we also have chronic inflammation. This is where immune cells have tried to do their job of healing, but unfortunately, there's some kind of ongoing stimulus that results in even more cell recruitment. This results in an increase in inflammation, and it actually changes the cells themselves, and it results in a repetitive cycle and an increase in disease. So this is where the problem comes in. But I'm going to look at acute inflammation because it is vital. If you understand acute inflammation, you can understand chronic. Okay, I'd like you to imagine that you're walking barefoot along a beautiful sunny beach, and yes, you're going to have to imagine sunshine today. And hidden in the sand is a rusty nail, which you unfortunately stand on. So that nail is going to pierce your first line of defense, which is your skin, and it's rusty, so it's going to have particles that you don't necessarily want to have in your body. And it's going to initiate what's called an innate immune response. This response is immediate, and it's modulated predominantly by histamine from mast cells, but there are cytokines and leukotrienes involved. This response is usually quite short-lived, but in the case of a rusty nail, it's probably going to be a tad longer than 15 to 30 minutes. So if in the case of your poor foot being pierced by a nail, we'd probably see the cytokine. So a cytokine is a chemical messenger, basically a mediator of sorts produced by your body to get the inflammatory response going and propagated and mediated. So we'll see TNF-alpha getting involved to prolong the cellular changes, which will eventually result in more white blood cells being recruited to assist in healing and repair. So in this case, we're going to need more white blood cells because who knows what else was on that rusty nail, other than rust, probably bacteria. I've mentioned trauma as a trigger. Infections are also, but we can actually initiate an acute inflammatory response to physical or chemical agents, so things like heavy metals, any kind of foreign body. I think of a splinter, but also other immune reactions. So you've had this trigger, then what happens? We get vascular changes. That's the one that we see quite obviously, because we have increased blood flow to the area, which causes the heat, pain, and swelling. But we also have cellular changes. We've got the recruitment of the white blood cells. The vascular response, this is initiated predominantly by mast cells and basophils. Those are the cells that contain histamine, histamine being a vasodilator, and it kind of helps the vessels become more porous so that the white blood cells can move out of the bloodstream and into the tissue. They'll move along with the fluid, so you'll get the swelling that happens. And the white blood cells and platelets would clean up the injurious agent and damaged cells and essentially stimulate that healing response. So the role of inflammation in this case is to stimulate the delivery and activation of white blood cells by chemotaxis. Chemotaxis is essentially the movement of the white blood cells to a stimulus of injury or infection or whichever. The neutrophils, they're fairly large. We have more neutrophils in our white blood cells than we have any other white blood cells. I think they take up about 60% of the white blood cell proportion. They are the kamikaze cells, so they will phage or eat with whatever they need to and essentially die and sacrifice themselves to become pus. Lovely, but true. Basophils, as I mentioned before, they're essentially the same as mast cells being in the tissue, basophils being in circulation. And they carry the additional heparin, which is an anticoagulant, and histamine, should the vascular changes that have been initiated need to be maintained. So if the inflammatory response needs to be extended a little bit. Eosinophils we see mostly in parasitic infections, but also in allergy-mediated inflammation. And the reason for this is that eosinophils' job is actually to subdue histamine and slow down or stop the vascular changes in the healing process. So a few days after stepping on that rusty nail, we have monocytes in circulation, which eventually become macrophages in the tissue. They are large phagocytic cells that arrive at the site of injury to eat the injured and damaged tissue or pathogens or whatever has been introduced to the body that shouldn't be there. They're essentially cleaning that area up. But in some cases, such as in TB, they can form fairly large giant cells and granulomas that in serious cases can be seen on X-ray. They're also responsible for secreting the inflammatory cytokines such as TNF-alpha and the interleukins. That's quite important to remember. So the white blood cells initiate that continuous inflammatory response. So if our initial wave of white blood cells hasn't managed to remove or deal with the bacteria and other infectious agents, then our next part of our immune system, our humoral system, kicks in and B and T lymphocytes will be recruited. They're fairly specific. So what I have here is just a little diagram to show you what a normal arterial and veneal look like. And you do see that there are a few of the resident white blood cells hovering around everywhere. But if in the case of an inflamed one, there is arterial dilation, expansion of the capillary bed, veneal dilation, increasing the blood flow, bringing more nutrients, bringing more white blood cells, and causing edema, which will help with the movement of the white blood cells into the tissue so that they can do their job and repair us, heal and repair. So I've mentioned mediators, chemical mediators, otherwise known as cytokines. These can be produced locally at the site of the infection or injury or are circulating in the plasma as inactive precursors that become activated at the site of injury. So we obviously don't want them active in circulation until they're absolutely needed. So what we have here, we have a stimulus being bacterial products or immune complexes, toxins, physical injury, or anything that could stimulate an immune response. These then activate macrophages along with other white blood cells, and TNF-alpha is the first inflammatory cytokine along with interleukin-1, but I'm not going to talk too much about interleukin-1. We'll focus on TNF-alpha. And you'll see that we have endothelial effects, systemic effects, fibroblast effects, and leukocyte effects. Interleukin-6, which is the other one I'll be mentioning a little bit later, is mostly involved in the endothelial effects, looking at procoagulant activity, a decrease in anticoagulant activity, increase in leukocyte adherence. And then if we go to the bottom, leukocyte effects, there is an increase in cytokine secretion of interleukin-6. So those are where the main players that I'm going to be discussing actually fit into the inflammatory response. Okay, so we've gone over acute inflammation, and hopefully you have a bit of an understanding. I'm sure most of you actually already knew that already, but let's consider what happens when an inflammatory response becomes prolonged. So I'm talking about it lasting weeks, months, even years in some unfortunate cases, and predominantly years that I see in my practice, during which this time inflammation, tissue injury, and the healing process all proceed simultaneously. So essentially, everything's happening at once, and there is no resolution and repair. So acute inflammation signs are very, very obvious. So there's the redness, swelling, loss of function, etc. That are those 5 pillars that I mentioned earlier. But the thing about chronic inflammation that I'm sure you're aware of is it's a little bit more insidious. Sure, they'll have chronic pain and frequent headaches, but low energy, chronic allergies, high blood pressure, and brain fog, a lot of practitioners wouldn't even think to check HSCRP and ESR in those cases to rule out inflammation at this stage. Brain fog being a particularly important one because nobody wants to lose their cognitive functions. I'm going to quickly go through the cells of chronic inflammation, but they're the same cells involved in acute. They're just doing their job a little bit longer. Macrophages, in this case, are the dominant cell of chronic inflammation. They're derived from monocytes, as I mentioned before. So monocytes are floating around in the bloodstream, and as they're triggered by some kind of stimulus, they will move out of the bloodstream and into the tissue in order to phage and clean up the areas. They are filters for particulate matter and microbes, any form of pathogen, essentially. So lymphocytes are our cell-mediated or humoral system, immune system cells, and they'll be involved as well in chronic inflammation. So if you do a blood test, you'll see in some cases an elevation of lymphocytes. Eosinophils, these are most often elevated in chronic cases of parasites or allergic inflammatory responses. I do like to check a full blood count whenever I'm looking at inflammation, because you do get a fairly good idea of what's going on, not just with the red blood cells, but the white blood cells will tell you what you're looking at with regards to the chronicity and the cause. So neutrophils, which are also phagocytes, they are the classic hallmarks of acute inflammation, but many forms of chronic inflammation continue to show extensive neutrophilic infiltrates. So quite important to check them out as well. Right, so I've mentioned the cytokines. And we have interleukin-6, TNF-alpha, and these 2 cytokines are elevated in, if not all, then most inflammatory states, and have been recognized as targets of therapeutic intervention, as I mentioned earlier, and please take the time to look at that research. There's another player on this slide, which I'm sure you've all noticed, methylene tetrahydrofolate reductase. not a cytokine per se, but because it is involved in inflammation, and I will go into that in detail a little bit later, we have included it in our tests. So our reports will include looking at IL-6, TNF-alpha, and MTHFR. Right, so let's take a look at interleukin-6. Just a note that we at myDNAhealth originally looked at interleukin-6 purely in relation to wellness and weight management. That was looking at our first panel that we ever developed, mostly because of its effect on glucose and lipid metabolism, and the fact that it's generally increased in obese and morbidly obese people. The reason for this is that it falls into the class of cytokines called adipose cytokines. So adipose cytokines, for those who don't know, are cytokines released by adipose tissue. So if there's more adipose tissue, there's more of a chance of chronic inflammation because these cytokines are consistently being released by the adipocytes. IL-6 decreases with weight loss, and it has a very interesting benefit from exercise in that it becomes anti-inflammatory and stimulates lipolysis or lipolysis. But there are more effects of interleukin-6, which I will discuss on the next slide. Again, I'm going to point out that there is a link at the bottom to some research that you might want to look at in your own time once we've completed this webinar. So we were looking specifically at the fat version of IL-6, but if you look at this, it's a main player in CRP. So if I'm looking to measure how much interleukin-6 is around, there are some labs that will allow you to measure IL-6 and TNF-alpha. So you're welcome to find those. But easy enough is to check CRP, C-reactive protein being produced in the liver in response to inflammation. The other thing that IL-6 does is it works with your immune system, working on neutrophil accumulation. There's loss of immune suppression, so we're looking at autoimmunity. We're looking at bone resorption, so people who have a predisposition to things like osteoporosis, it's a very important player there. We're looking at the matrix deposition of collagen and fibroblast deposition, not so great if you're doing that in your arteries. And we're looking at tissue destruction ultimately. So more than just I'm overweight and producing an inflammatory response, there's quite a lot of morbidity in this particular cytokine. So as I mentioned, it plays a big role in inflammation in general, and specifically regulates the expression of CRP. So we know that if we're looking at somebody where we suspect any kind of inflammatory response, you can measure CRP or the high sensitive version of it, or you can look at ESR and ferritin. Ferritin is also an inflammatory marker. The chronic inflammation that we're looking at is often associated with obesity and visceral fat deposition. There's a tendency to insulin resistance, dyslipidemia, and there is an elevated risk for cardiovascular disease, but also bear in mind things like osteoporosis and autoimmunity. The beauty of this particular genetic variant, because I'm going to be speaking in genes now, so interleukin-6 is made by the gene IL-6. If somebody has a SNP at this particular gene, they need to be encouraged to exercise. So exercise being clinically proven to improve the expression of this gene. Obviously, people who have a fully expressing IL-6 will be in pain because they're inflamed. So offer them suggestions like weight-bearing exercises that they can do just sitting down to start off with, or cycling or swimming. Any kind of movement is better than none in this case. So we've got our reports that will tell you whether a person has the wild type, which is the average of the population, and an AA version will come up as no impact. The risk allele is the C allele, and that's been associated with an elevation of IL-6 and CRP concentrations. And as mentioned before, it's related and associated with inflammation, obesity, insulin resistance, dyslipidemia, and hypertension. Very importantly, this particular genetic variant is made worse by smoking. Many of them are, and I'm fairly sure that all of us in practice do kind of have a stern word with the people who choose to smoke. But if you do have a smoker who has the risk allele in this particular variant, just point out to them that they are essentially making themselves sick every time they take a drag of their cigarette. Nutritionally speaking, we encourage people with this allele, the C allele, to follow a diet that's rich in antioxidants and high in omega-3 being specifically anti-inflammatory. So I would encourage my people to eat a rainbow every day, really get their nutrients through diet first, possibly supplementing the omega-3. It really depends on what their questionnaire results have said, because if their questionnaire results are telling me that they're actually doing okay, then maybe supplementation isn't even needed. So let's look at TNF-alpha. This is one that's particularly linked to adipose tissue. There is a direct correlation between your body size, your BMI, and increased obesity, and the expression of TNF-alpha. Interestingly enough, it is also suppressed by exercise. So again, any variant in this particular gene, I'm going to be recommending that they get some kind of exercise. As I mentioned, it is an adipocytokine, but it's also expressed by macrophages. So if somebody has a long-term underlying infection of some sort, maybe one that you can't even see, like periodontal or in the gut somewhere, they'll also be expressing TNF-alpha more than somebody who is lean and healthy. There is a direct link between this particular marker and arterial inflammation and diabetes. With TNF-alpha, on the left-hand side of this slide, we have things that improve the expression. In other words, they can have SNPs in this area, but exercise and certain dietary supplements can downregulate the expression. But overeating, smoking, aging, not exercising will actually upregulate the expression of this and predispose people to things like vascular inflammation, thrombosis, and atherosclerosis. In our reports, we have the GG variant having no impact, and the A allele increases the risk of an upregulation in the expression of TNF-alpha, and that can predispose the person to arterial inflammation, insulin resistance, type 2 diabetes. If this person is expressing TNF-alpha, they will probably have an increase in CRP levels. So if you want to do functional tests to take a look at whether they are indeed expressing this or not, have a look at the CRP, my preference being HSCRP, but any CRP will do. And in their case, sugar is actually a pro-inflammatory nutrient. So they need to have a diet that is low in sugar and obviously focus on getting their nutrients from really good sources. So we've covered the cytokines, but now I've added some random SNP that obviously doesn't look like it's a cytokine, but it is the main player in methylation. And if you can see on the slide, you'll see that there is a link to an inflamed gut. There's not just heart disease and neural tube defects, there's cancer, Alzheimer's, yeah, quite a few. Go through the slide when you have a moment and you'll see migraines are on here. So a mediator of inflammation, definitely, not a cytokine of inflammation. So MTHFR, if you have a risk allele there, if you have a mutation in that particular part of your folate cycle, it essentially stops your body from converting homocysteine to back to methionine. And elevated levels of homocysteine have been linked to arteries doing sudden contractions. So there's often sudden death from cardiac events that aren't necessarily related to things like atherosclerosis. So you can have somebody dying quite suddenly, and when they have the autopsy done, there's no real sign as to why, but yet they have this elevated homocysteine. It's also a pro-inflammatory molecule that really does need to be methylated to improve our antioxidants as well. So yeah, again, there is a link at the bottom, so you can have a look to see how MTHFR really goes deeply into inflammation, particularly, and looking. I'm focusing on cardiovascular disease in this case, but any kind, cancer, migraines, inflammation in general. So in our reports, if somebody comes up with a CC variant, that's excellent. I'm looking specifically at MTHFR 677. There will be no impact if they have the Cs. It's only when they have a T. If they are a CT, it means that they have about a 30% reduction in their ability to methylate. And if they have homozygous TT, then they're going to have only 30% functioning. So in other words, a reduction of 70% in the ability to methylate properly. Sounds worse than it is, because I'm just going to go back here. It's very easily looked after when you're eating really good folate sources. So a diet rich in leafy green vegetables and high in choline and things like that, supporting methylation through diet would be a key factor here. So not everybody who has this particular variant is going to need to maybe take a supplement. If they're healthy and looking after their diet and avoiding toxins and supporting phase 2 detoxification, they'll be okay. And we can turn on and off the particular genes through our diet and lifestyle. So this one in particular, where unfortunately when it first came to be in our field, everybody who had the particular variant immediately got put onto methylation factors, and that's not really advisable, because you can over-methylate, cause more problems in the long run. So over-methylation would be symptoms like anxiety and joint pain. So don't always treat a SNP just because it's there. Have a look at the expression. The expression is whether that gene is turned on and causing problems or turned off and not, as a brief summary of what that is. So we don't just look at the genes. I think I've mentioned this in quite a few of the webinars that I've done. Just having the potential doesn't mean that you've got all the risks happening or expressing at that moment in time. So myself, I have homozygous MTHFR, and when I wasn't well, having massive exposure to toxins and being stressed and not sleeping well, that MTHFR was expressing freely, and I had plenty of problems I'm not going to bore you with today. But nowadays, because I live a healthy life, I sleep well, I look after myself, I don't have any of the issues. I don't have a crazy inflammatory response, which I will talk about again a little bit later, just to give you some, personal experience as well. So having the variant, having the potential doesn't necessarily mean that it's actually affecting your health. So the way we at myDNAhealth look at this is we get people to fill out subjective questionnaires to get their perspective on how they're feeling and whether the genes are expressing. Just a note here, if you get one of your clients to do the questionnaire and the results kind of don't match what you're seeing, get them to redo it, because people do kind of downplay sometimes their symptoms. So if things are not gelling with you as a practitioner, please, by all means, these questionnaires can be repeated. If things are not looking great to you, get the person to do it again. I find that quite often in stress. So a person will tell me in the questionnaire that they're absolutely fine, but when I talk to them in consultation, there's a lot of stress happening there, and I'll go through the questionnaire with them. I'll retake it and get them to give me a more honest version. So essentially what I'm saying is, if they're coming up with a healthy questionnaire, I'm just going to encourage them to continue with their healthy habits. I'm not necessarily going to put them onto an anti-inflammatory diet or anything like that. I'll point out if they have risks, I'll point out their risks to them to get them to maintain their healthy lifestyle, but not necessarily treat. I only look at treating the ones that need to be treated. So if we look at this particular set of results here, this is someone who I'm probably going to talk to about their lifestyle, their habits. Their questionnaire results have come up with unhealthy inflammation. And in the questionnaire, if you haven't seen it yet, we talk not just only about the pain that people might be feeling, but also their predisposing factors according to diet. So if they're coming up as eating lots of inflammatory foods, they'll get a long lecture and some handouts on what is a less inflammatory diet. In this case, the person has homozygous interleukin-6 and MTHFR. So I'm going to be instructing them to eat lots of leafy greens, and in the case of interleukin-6, lots of colorful vegetables, get their sources of omega-3s in, and encourage a little bit of exercise. Really, depending on their symptoms, if this person is able to walk or cycle, that would be ideal. If they're in so much pain that they can barely even move, then just some weight-bearing exercise to start off with. Slowly but surely, as they improve, I will increase their exercise. Okay, so my point being is that we don't just look at the genetic predispositions. We obviously, most of us have predispositions to something, but it's whether those non-self external environmental factors are influencing our gene expression or not. I've mentioned exercise a few times, nutrition as well, environment. So I'm going to talk a bit about pollution. Very important is emotional health, and I'm going to cite some research about that in a minute. So essentially, we get given this information, and it's thereafter our choices that determine whether a gene is turned on or off and causing harm or not. So let's look at some of the influences. Injury, obvious. Infection, another obvious one. I've kind of covered that, but I'm going to go into more detail. Inflammatory biotoxins. This is a big subject these days. And biotoxins can include things like Lyme disease or Epstein-Barr virus. In my case, I've had mold before, and it's very pro-inflammatory. Sleep. Many people don't realize how important sleep is. And in this day and age, there seems to be a trend to thriving on coffee and no sleep, and it's detrimental to a lot of people's health. Our thoughts play a role in inflammation, and I'm going to tell you all about that in a second. So stress would fall under that. Obviously, our food and pollution. So what we're exposed to through air, water, pesticides, persistent organic pollutants, etc. So injury is fairly obvious. I'm not going to spend too much time here. I kind of discussed the rusty nail. But if you consider, in that case, you're piercing your skin and you're introducing iron and oxide molecules and potentially bacteria. But if you consider, say, a sprained ankle, no foreign bodies are involved, but the same vascular and cellular events will take place to precipitate healing. I've recently recovered from sprained ankles, so I'm going to skip over to the next slide just to ease my mind. Pollution. There's been a major increase in things like autoimmune conditions and chronic degenerative conditions over the past less than 50 years. And if we look at the pollution that we've been exposed to, about 70 years ago, but most of the pollutants that we're exposed to on a daily basis didn't even exist. And studies have shown that the average woman, for example, before she's even had breakfast, will have applied over 100 different persistent organic pollutants onto her skin. So quite a lot of exposure there. People don't filter their water, they don't wash their fruit and veg, they eat pesticide-ridden foods. So I'm going to cite the research at the bottom of the slide for you. The take home from this research is whether pollution can cause inflammation, specifically affecting cardiovascular disease. And their results show there's an immediate response to particle number concentrations on the interleukin-6 level specifically, possibly leading to the production of acute phase proteins as seen in increased fibrinogen levels. That's going to increase the clottingness of the blood and increase cardiovascular risk. They're saying that there is a link between air pollution and adverse cardiac events. In the case of being exposed to pollution, I personally own an air purifier. I purify all the water I drink, and that is the advice that I would give to people who have a potential to be inflamed. And say, for example, I live in London, so my exposure is pretty high, and yeah, an air purifier while I sleep definitely helps. And we have the biotoxins. Mold has been linked to something called the chronic inflammatory response syndrome, or CIRS, but not just mold. There's a strong link between viruses and bacteria. And Chris Kresser says that sufficient exposure to biotoxins causes a priming inflammatory event. In other words, that's something that triggers and activates the immune system, such as a serious upper respiratory tract infection or even Lyme disease, which is becoming more prevalent these days. So if your client is not doing well and is constantly inflamed, maybe consider their house as a toxicant and check to see if they've got any kind of mold. And mold is tricky because you can't always see it, but you'd be looking for damp specifically. In this case, if you're working on nutrition and lifestyle, you probably won't get the solutions that you're looking for. We as practitioners nowadays need to look not just at the person and what they're putting into their bodies and excreting. It's what environment are they living in, and can they fix it, or do they have to move? I've had the case where I myself had to move because of the amazing amount of mold I was being exposed to every day. Right, sleep. I've got a number of research articles on the importance of sleep. The first states that loss of sleep, even for a few short hours during the night, can prompt the immune system to turn against healthy tissues and organs. Losing sleep for even one part of the night can trigger the key cellular pathways that produce tissue-damaging inflammation. The findings suggest a good night's sleep can ease the risk of both heart disease and autoimmune diseases such as rheumatoid arthritis. And you'll notice that when you run a test through myDNAhealth, we do add a sleep questionnaire in our comprehensive, because sleep is very important for many things, for methylation as well, for repair of all of our tissues, our brain included. The second research that I'm going to cite with sleep is sleep disturbance is associated with inflammatory disease risk and all-cause mortality. That's a massive statement. Here they've assessed global evidence linking sleep disturbances, sleep duration, and inflammation in adult humans. Both of these are cited on the slide, and you're welcome to have a look at them. A book that I encourage my patients who refuse to listen to me when I tell them that sleep is important. I tell them to get Arianna Huffington's book, The Sleep Revolution. And I also cite one of her references is that not sleeping well can activate over 700 pro-inflammatory genes. And if you think that we're only looking at 3 and there are many, many, many other pro-inflammatory genes, just sleeping will turn those genes off. That's how important sleep is. So thoughts. A few years ago, I had chronic inflammation. I unfortunately had an injury and I was living in a moldy house and I had MTHFR. So my HSCRP was hovering around 13 and I thought I was doing everything right. I moved home and I was supporting methylation, but I just wasn't able to get this CRP down, and I was kind of becoming obsessed about it. I'm luckily associated with a couple of people who are professors of pathology, and I emailed one of my friends, and her response to me was no words except the link to the research that I've actually cited below. It's titled, the differential effects of post-stressor rumination and destruction on cortisol and C-reactive protein. Basically, what she was pointing out to me, that my overthinking about my elevated CRP was actually affecting my stress and CRP levels. This research shows that meditation and other such distractions, so not thinking about the problem, can lower the CRP. And I'm pleased to say nowadays, no problem with my CRP. The emerging role of chronic inflammation in the major degenerative diseases in modern society has stimulated research on the influence of nutrition and dietary patterns on inflammatory indices. Most of the studies correlated the analysis of habitual dietary intake. In other words, we're eating the same 30 foods every day. This is determined by a frequency questionnaire or a 24-hour recall. They would take that information and look at systemic markers of inflammation like hsCRP. There have been several controlled interventions which evaluated the effect of the change in dietary pattern or of certain foods on inflammatory markers in defined populations, and most studies reveal an effect of dietary composition on some of the inflammatory markers. The most well-studied one is a traditional Mediterranean dietary pattern, which typically has a high ratio of monounsaturated fats to saturated fats with high omega-3 versus omega-6 polyunsaturated fatty acids. It supplies an abundance of fruit and vegetables, some legumes, and a few whole grains. Has been shown to be anti-inflammatory, but there are other anti-inflammatory diets, and please always take into account the fact that a lot of our patients do have leaky gut and dysbiosis, which are both going to play a role in inflammation. So have a look at gluten, dairy, and in the case of TNF-alpha, sugar, if they have ongoing chronic inflammation. So, we always say gut first, so this should have been my first slide, but it is not my first slide. It is the one that I'm going to essentially end my webinar with. So what can you do as a practitioner? You can certainly check for predispositions genetically. And if they have the predispositions, but they're telling you they're okay, that's fine too. You don't need to be treating something if it's not expressing. If you can, remove the source of the inflammation. Often supporting methylation and detoxification will help with inflammation. Meditation and sleep, very important. Assisting with diet, and in some cases where necessary, supplements, and encourage exercise. So really look at all the multifactorial influences of inflammation. So I'm going to hand over to Sylvia, who's going to help you out with this quick poll. Sylvia?
Hi, thank you. That was a very informative presentation. I think there was a lot of useful information in there. So to run the poll, if someone has inflammatory SNPs but an apparently healthy lifestyle, would those SNPs still need to be treated? So tell us what you think, yes, maybe, or no. Okay, I can see the results coming in. Thank you for that. That's very interesting. Great. So we have a few questions from our practitioners, actually. So we'll move on to those. And following from that poll, let's start with this one. Can we look at the DNA results alone and determine whether the person is actively inflamed?
Can you look at the DNA results alone and determine whether they're actively inflamed? No. When you're looking at the DNA results, you're looking at their predispositions, their potential. But if you are wanting to find out if they're actively inflamed, you can either run labs or you can ask. Some people will tell you that they've either got pain or stiff shoulders, sluggish brain, all of the things that I mentioned earlier, the brain fog. But ideally, you want to be looking at labs, hsCRP and ESR and ferritin. So no, you can't tell whether a person is in active inflammation just by looking at the SNPs.
Great, thank you for that. So when you talk about exercise, what do you mean by improve the expression of a certain SNP? If an increased expression is negative, then doesn't that mean exercise will downregulate the expression?
Exercise would down-regulate the expression. That's exactly it. So do you want to repeat that question?
Yeah, that's great. I think that's clear now. And
Okay.
we actually have two questions related to mold. So I think, given your experience, you can answer.
Absolutely.
So, how do you find out if you have mold in the house?
Oh, okay. Unfortunately, mold, if you can't see it, doesn't mean it's not there. Generally, you look for areas of damp or a history of damp in your house. I must admit, though, that I see quite a lot of mold in people's houses, and they're just not aware of what it is and how damaging it is. We live in a fairly moldy environment in this country because there is so much water falling out the sky all the time. So I'd say the majority of homes have a risk of having some kind of mold in them. Take a look at how quickly black mold arrives in your bathroom. Make sure that you're constantly cleaning it out. I actually use, because of my exposure, once you've been exposed to mold once, you will always be a lot more sensitive to it. So you can walk through a moldy building and you'll start to have a reaction. But the Dave Asprey has developed a product called Homebiotic, which I use in my living environment that I'm in mostly. It's probiotics that help to regulate mold in the house. Fantastic. Unfortunately, I'm not too sure if it's easily available in this country. I am trying to get it into the country because I would rather treat my environment naturally as well. So using probiotics to combat the mold. But yeah, there is no real way of measuring how much mold you have in your house. If you can smell it without seeing it, you've already been exposed. So in the house that I was exposed in, I had a particular cupboard that every time I opened it, I could smell the mold, but I couldn't see it, but I was still being affected by it. So mold is a tricky one. Look more at the dampness of the environment, because if the environment is damp, that's a favorable environment for mold.
Great, thank you for that. And the other question was, does mold cause a problem if you have wild type of MTHFR?
It causes a problem if you have wild type MTHFR that's expressing, no. Okay, so wild type being CC. There's more to mold than just MTHFR. There's specific, the type of DNA testing that they look at for bone marrow biopsies. So we look at that kind, it's HLA typing. If you have a specific HLA type, which we don't test, it's a very complicated lab test. It has to be done elsewhere. Then your predisposition to becoming sick from mold is much higher than, say, a person who doesn't have that type. So you can have two people living in the same house, and the one person gets really, really sick on any exposure to mold, and the other person doesn't. And that's a much deeper genetic cause, so not one that we cover. So if somebody has an MTHFR 677 wild type, but is genetically the type of person who is susceptible to mold problems, it doesn't really matter that they're CC MTHFR, they're still going to be affected by it. And again, if somebody doesn't have that HLA typing and they're living in a moldy environment, they're not going to be as affected by somebody who does have it. The big subject. I'm probably going to have to do a webinar on it now, aren't I?
Probably, that would be interesting. So thank you, Shania, for that. I think we have time for two more questions. So someone was asking, if someone has a moderate or severe questionnaires result, but the SNPs are all good, do we just ignore the questionnaires result?
No, not at all, because they're telling you they're inflamed. So have a look at what else could be contributing to their inflammation. We're just looking at 3 of the main players in inflammation. There are other ones. So have a look then at their diet, lifestyle, environment, exercise. Have a look at what they're putting into their bodies that could be precipitating an inflammatory response regardless of their genes.
Okay, and the last question we have is, what if someone is inflamed and is being asked to exercise, which may make the inflammation worse? How do you then encourage them to exercise?
Okay, so ideally, if I'm demonstrating it to myself. So if you hold out your arm and contract all your muscles all at once and hold that for about 5 seconds, that is exercise. So if somebody is completely inflamed, can't even put their feet on the ground, and I've treated many people like this, so there are cases where this is definitely the type of exercise that they can do. It's not putting any strain on anything. It's holding a flexed muscle for about 5 seconds. And then introducing weights. So it's weight bearing eventually. And as they slowly but surely get stronger, because that exercise itself is going to down regulate some of the inflammatory processes as well, eventually they'll be able to start moving slowly. And you do it very slowly. You don't say, if they're bed bound, you don't tell them to get up and start moving. Not at all. You can actually just do that static, as long as they're contracting their muscles, they are actually getting some form of exercise. And as they start to improve, you can include things like slow walking, and then cycling, swimming being fantastic, because then it's hydrotherapy as well.
Great. Thank you, Shania, for your answers and for your great presentation. And also thank you for your questions. And thanks for attending. So we will be sending out the webinar recording along with the slides, so you'll be able to actually look at the research and along with your questions and answers. So that will be reaching you by tomorrow. So thank you again for attending. We hope to see you in our future events, and that's where we'll end. Thanks again.
Bye.